There's over 15,000 diseases. Our mission is to accelerate drug discovery.
AI has impacted almost every area of medicine.
Faster in discovering a medicine, faster in developing it.
What we're on the cusp of is perhaps the most powerful technology force of our time.
Drug discovery is being disrupted. Emergent technologies alongside artificial intelligence have the potential to transform how we understand disease and develop new drugs. But how are these advancements truly impacting the biopharmaceutical industry and the people and processes within it? I'm Kelsey McCloche, head of AI solutions for health and life sciences at Dataiku, and I'm reaching out to experts across the field to understand exactly what that impact looks like.
Often it takes 10 years, 12 years to get a new molecule to the market. That often starts with 10,000 different molecules that they're experimenting with, thousands upon thousands of manual experimentation processes.
I am probably characterized as an industry veteran, 35 plus years in the pharmaceutical industry in various roles. If I think back to when I started my career, I started in a large pharmaceutical lab that basically was still, stuck in the 70s and 80s. So we were doing things like printing out tables of data, cutting that out and gluing it into a paper lab notebook. It's not a good way to create value from data.
When I have an AI tool at my fingertips, I effectively have hundreds or thousands of workers, if you will, that can go do work on my behalf.
There's this article that came out of MIT where this lab built a model to extract atoms, bonds, and the layout of the entire compound directly from an image. In this patent specifically, there's 678 compounds. So that's a lot of work for somebody, right? So if I can speed up the process by going through it with a machine and do that automatically and then have the human verify, that's a much easier process.
It's not that computational biologists or researchers or radiologists are going to be replaced. It's that they're going to become far more productive, both within their own organization and for society at large.
In the past 15 years, there's been a rapid acceleration of bringing the cost to sequence DNA down, which has enabled us to generate data faster than ever before. It's actually now called genomical speed instead of astronomical speed because we've already eclipsed all the data that's ever been made by astronomy with genomic data. So what that means is something that used to take $100 billion to do once, for example, sequencing one human genome, instead of years, we can do it in a matter of hours, and instead of hundreds of billions of dollars, we can do it for $300. So it is really an extraordinary and precipitous drop in the cost, which enables us to make data faster and gives that much more data for our algorithms to learn from and to build models on.
Is AI really capable of tackling complex biological processes that lead to disease? Can we find those disruptions and design new molecules that act upon them before we even get to the lab? AlphaFold, an AI system developed by DeepMind, was groundbreaking in its ability to crack the code of protein folding with remarkable accuracy, predicting the functional form of over 200 million proteins and unlocking a new foundation for computer-aided drug design.
One of the most amazing breakthroughs that I've seen recently has been the ability to predict protein structure from primary sequences. And so AlphaFold, from its very first breakthroughs about 3 years ago, through even more recently, some of the newer developments in terms of being able to predict protein ligand structures and really kind of expand on the scope of what was possible earlier, is very real and has tremendous potential.
We have been able to synthesize information faster, find interacting proteins that we were looking for before you do it manually. Now you can find every possibly interacting protein to your target and then match that and use that for designing a drug.
I'm Dr. Shahar Keenan, co-founder of Polaris Quantum Biotech. We use technology to design novel molecular drugs. AlphaFold, in many cases, is where we start. You usually design new molecules to fit a protein pocket. You either activate the protein or you block the activity of that protein. For a molecule to become a drug, it needs to have many properties, starting from, of course, the biological activity, either stopping a disease or slowing a disease, how easy it is to synthesize, toxicity, side effects. You're looking for many properties for that single molecule. Looking at very large number of molecules and finding the ones that have all the properties you want very fast, this is what we do. And we use quantum computers to do that.
What exactly are those objectives here, and how do they work with the molecule we're looking at?
We can say what we want is a molecule that will be similar to that one, but better than it. So this would be a best in class. Or we can say, no. We want a molecule that is completely new, but at the same protein pocket. That would be a first in class.
It's fascinating.
We really made a bet the company moment on this concept of deep learning, and we made sure that every piece of AI code had NVIDIA code built into it. And so that underlying hardware technology that was really good at rendering a video game was actually incredibly well suited for the math and deep learning as well. This is an area that we spent a lot of time working with the developers on, making sure that we could take something that would take, call it 10 weeks, and bring it down to 2 days.
I'm Kyle Trettina. I'm an alliance manager for In Silico Medicine, where we develop our own assets and drugs. Our algorithm is not constrained by what exists, essentially. You go into it with a target in mind, you hand it the target, you then will offer it the profile of the molecule that you want in terms of novelty, safety, metabolic stability, and other factors, and it will start to imagine molecules that fit into that space.
Who would be the user for this, and how's it changing their usual work?
Good question. So the user that we had first in mind is the biologist. And specifically those that maybe don't have a very strong programming background, but still want to use multi-omics data thoroughly in all of their decisions about which targets to select, what genes to screen in the various assays. You can filter by expression in different tissues. You can filter by a certain gene set. I find that pretty much every scientist wants to define their own set themselves by their own experiment.
It's not AI telling you this is your result and this is what you should go do. It's allowing these scientists to put their own intelligence into a system so that AI is also aiding and assisting them, but they're also still adding their own human intelligence into it.
Yep, that's exactly it.
By analyzing vast data sets at unprecedented speeds, AI can rapidly accelerate the traditional scientific cycle of hypothesis and experimentation. So how do you move from prediction to reality, from a computer to a lab? How can we ensure new AI-augmented processes will be trusted?
There is a lot of hallucination with artificial intelligence. Even if you go back to your regular OpenAI and you ask for feedback on summarization of a blog, or you want to write a letter and you ask for a summary, there is some aspect of it that is not real. There is some aspect of it that doesn't make sense. And that's the same thing when you use GenAI for R&D. You could ask AI to generate a new protein sequence or a new viral variant sequence. There is always a caveat that it might not be something that you see in reality. It may not be something that you will ever find in a lab, or you can even create in a lab. It may not be stable. So there is always a subtext there that says AI is a potential. To make it real, you still have to go back and verify all of this, right? So to me, the timeline can only be accelerated once we have done this enough number of times that we can actually figure out all of those things that are not real and refine our algorithm to make it more accurate.
When it comes to small molecule design, the situation is a lot more, I would say, optimistic in the short term, because we can validate these in the lab pretty much right away. Our algorithms only take minutes to hours to imagine all the molecules that you need for your experiments.
The computational result can be validated in the wet lab with a very high success rate. So basically, it takes two teams, the computational team and wet lab team. All the data has to flow between the two teams to fully utilize. We use data to generate 50 antibodies. 8 of them are effective, so the hit rate you can see is pretty high.
One thing is clear, AI systems cannot work in a vacuum. For real and lasting impact, it will require data scientists and bench scientists to work together. Realized success may take a long time, and the ability to fail fast may be the earliest evidence of it.
The first three molecules invented by AI have all now failed in further downstream testing. So, this is still a very complex set of processes that all have to come together to invent a new medicine.
Algorithms will always get better. There will always be new tools, new computational methods that get more and more refined. But at the end of the day, if you want to know two physical objects, do they interact with each other, or does this one drug lead to a better treatment, you have to test it in people, in cells, at the bench. But the joy is, what if the algorithm is 99% of the way there? And then you've just confirmed your prediction. So that's what's great, is you can confirm what you think you know right at the bench.
If we can start predicting and triaging and prioritizing the right experiments to perform, then we're going to speed up that entire process. And that's what's going to make medicines more available, more accessible, and come to market faster.